So the next one is filtering improves the biologic, and spicules, and what are spicules? So spicules are the soft tissue that resides between the hard bone. Spicules are soft tissue. And when a hematologist does a marrow aspirate to check for any kind of blood cancer, the first thing they do is they put a drop of the marrow on a glass slide. They put another slide on the top. They smear the two together. Then they hold up the slide and they look to see if there's any spicules that they can see with their naked eye. And if there are no spicules, then it's reported as a blood tap. Because if there's no spicules, I didn't get any marrow. The other thing about the spicules, when they put the glass slide on top of them, they say, don't press the glass slide. Just the weight of the slide alone will flatten out the spicules. So it's not pieces of bone. I think sometimes people get this impression that spicules are pieces of bone. There's no way the weight of something that weighs, less than half an ounce is going to crush bone. So it is soft tissue. And, what this paper here shows you is what are spicules, and then, of course, all the other things we just talked about. You do a big draw, you're going to get a lot of peripheral blood, and if you don't have any spicules, then you got nothing but peripheral blood. So spicules are soft tissue. They are not hard bone. So this is an aspirate, and then this is the concentrate down here. So aspirate and concentrate. The same number of spicules, pre and post filtering. And the middle one is drawing from the sides as you stage the needle out. So notice, very high cellularity when I draw it that way, and a lot of spicules, which means it's a very high quality aspirate. No spicules means you have a very poor aspirate. And this work was done by Dr. Mandel at Harvard, and these patients were spine fusion cases. We did a comparison of our product when we filter and we don't filter. We had a drop-off in total nucleated cells. We also had a big drop-off in CFUs. So we went from 1,300 to 600. So like a 54% drop-off as a result of filtering. So we don't feel filtering helps. And the reason that centrifuge companies filter, they have to filter in order to make the centrifuge work. Has nothing to do with a clinical reason. And that's something to keep in mind when someone says, you need to filter because everyone else is filtering. They're filtering because they're centrifuging. You have to filter when you centrifuge. That ends on spicules. Do we have any questions on that, Nick?
Absolutely. Why do centrifuge companies filter pre-centrifuge and not post-centrifuge?
That's an excellent observation. In that, think about that. If I'm worried about spicules and I'm a centrifuge company, why wouldn't I run the whole thing, get my biologic, then filter it? Because now I'm sure I filtered right before I put it in the body. They don't do that. They filter, then put it in the centrifuge. They do that because they have to, or the centrifuge doesn't work correctly because of the mechanics of how that blood separates in the centrifuge. So if they were really concerned about spicules, they'd run it, then filter it. So filtering within a centrifuge product is due to making the product run more consistently. And I know that when Harvest was the first company, I used to work at Harvest, to launch a bone marrow centrifuge system. And when we first launched it, we had very inconsistent percent recoveries. And we said, why is that? And part of it was the fat content and the red cell content impacted how things sedimented and also impacted the way the shelf floated. When we filtered it, we eliminated those variables and we got much more consistent recoveries. Had no clinical reason other than it made the centrifuge work better. Then other people copied it and got the same result. Their results, their consistency of the centrifuge products started working better when they filtered it. No clinical reason. And then some people say, well, spicules are bad. That's crazy. If you don't have spicules, you didn't do a marrow aspirate. That's your test to show that you have marrow. And ask any hematologist. They'll tell you the first thing I do is check for spicules.
Absolutely. So, Andy, just in closing with the myth number 3, if you don't have spicules in your aspirate, you have peripheral blood, not marrow-derived blood, correct?
Absolutely correct. And this paper here So this is from a hematology text. In the absence of particles, which would then also mean absence of megakaryocytes or other hemopoietic precursors, the sample should be reported as a blood tap, peripheral blood. Don't filter out your spicules. That makes no sense. And don't centrifuge away your entire red cells and by default all of your stem cells. Makes no sense.
And can we remind the listeners that the marrow space is made up of trabecular bone? When we're doing the aspirate, we don't start drawing until we're through cortical bone. Once we're through cortex, we put a different aspiration tip on, and now we're drawing through trabecular bone.
That's right. And you think of that honeycomb trabecular bone. It's connected in a bunch of places. So when I break through it, pieces aren't coming off. It's connected, all on the other sides. What you're pulling when you're seeing those spicules is the pieces of the soft tissue, the true spongy marrow that's in between those crevices. And the reason hematologists are most interested in that's what I have to do a histology on to see if this person is really sick. I don't test hard bone. I test the engine of your life, which is that marrow between those hard bones.
Well done.
Make sense?
Absolutely. Well done.